Right, good afternoon, everyone. I am Rashmi Kanagal. I am an associate professor in hematopathology and molecular diagnostics at MD Anderson Cancer Center. I also serve as the director of the microarray facility at the CLIA certified molecular diagnostic lab at MD Anderson Cancer Center. And I will be talking to you today about our experience in performing whole genome optical mapping using Sapphire system in leukemia samples. Now, we're all aware that the knowledge of structural variants is very important both for diagnosis and prognosis in hematologic malignancies. And in some cases, identification of structural variants can alter therapy. Unfortunately, at this time, the high throughput technologies that have not been developed in order to identify these structural variants. Unlike the point mutations where identification has been revolutionized by introduction of NGS. Now, the structure variants includes both the copy number changes as well as balanced rearrangements. And right now, the identification of both of these variants is limited by the need for use of a combination of techniques. And this would include the conventional karyotyping, FISH studies for known aberrations, as well as microarray studies. The NGS studies for identification of structure variants have not reached the mainstream yet. Since Sapphire is able to provide this opportunity by using optical mapping, it can, as a single platform to identify these, we believe that there may be a lot of potential in profiling leukemias. Now, Sven has already mentioned, he described the technicalities of this system. So I am going to actually talk about the clinical utility or application of Sapphire into profiling leukemias, specifically in a disease called Myelodysplastic Syndrome. And the reason we chose Myelodysplastic Syndrome was because the structure variants have a big role to play in determining patient treatment. So just to give a brief overview, myelodysplastic syndromes are clonal neoplasms. They are characterized by three things. So firstly, they are characterized by cytopenias due to ineffective hematopoiesis. Secondly, they have a lot of dysplasia in the hematopoietic cells. And thirdly, these patients have a very high risk of transformation to acute myeloid leukemia. In fact, first looking at this MDS would also give us an opportunity to understand the transformation, the clonal evolution that's involved in transformation to acute myeloid leukemia, because a significant proportion of AML arises from patients with prior MDS. So MDS is, nearly half the MDS is characterized by structure variants, and I'm talking about de novo MDS in patients who have not received chemotherapy or radiation for any other cancer. And if the patient has had a history of chemotherapy for, say, breast cancer, we call it as therapy-related MDS. And in those patients, nearly 90% of patients have a structure variant. So the diagnosis of MDS, based on the WHO guidelines, the World Health Organization guidelines right now, is includes recognition of dysplasia. So we need, based on the criteria, we need to identify dysplasia in at least 10% of cells in at least one of the cell lineages, either erythroid, granulocytic, or megakaryocytic lineage. In some cases, especially if the dysplasia is very subtle, it is very hard to make this diagnosis. And this is even harder if the specimen, the bone marrow aspirate, is of a limited quality, which can happen with the associated fibrosis. So in such cases, if we are able to demonstrate a structural variant, that can actually help the pathologist push towards the diagnosis of MDS. So in fact, the WHO has issued the following listed abnormalities, both balanced translocations as well as unbalanced abnormalities. They've listed these as, they call it as presumptively diagnostic for MDS in any patients with cytopenia. So if we are able to, if we have a patient with unexplained cytopenias and we are not able to characterize the dysplasia, but we see one of these abnormalities, it's an automatic diagnosis of MDS and he needs therapy. Secondly, the structure variant, the type of the structure variant can actually characterize distinct types of MDSs, and these are biologically different. For example, MDS with isolated deletion 5q, and by isolated deletion 5q, it has to be either one abnormality associated with deletion 5q or no abnormality. These patients require actually respond very well to a type of therapy called lenalidomide. So it's important to pick these up. And at the same time, if the del5q occurs in a setting of a complex karyotype, of course, those patients do not respond. So it's really important to identify all of the aberrations in MDS. But even more important in MDS is because of the risk stratification. So I'm going to talk a little bit about that. So in MDS, there's this big study that's published in 2012 by major institutions. It's an international study by cytogenetic experts where they've characterized 5 separate categories of cytogenetic abnormalities depending on the outcomes in MDS, as we can see to the right. So what we see here, the outcome can range from very good to very poor, and this is dependent on not just the number of the structure variants, but also the type of the structure variant. And these also include translocations as well, not just the copy number changes. And so this category plays a very important, it's actually a primary component for what we call a revised IPSS risk score. So this is a universal risk scoring system that every oncologist uses for managing MDS patients across the world. So basically, depending on the cytopenias and other clinical parameters, as well as the cytogenetic category, the patient is assigned a score, and the treatment decision is made. Firstly, does this patient need the treatment? And secondly, what type of treatment? And all of the decision is made based on the IPSS risk score. And as we can see, this cytogenetics or the structure variance is one of the primary components. So MDS, it's a really good disease to understand the role or the utility of optical mapping in MDS. Unfortunately, even at this time, the cytogenetics right now is assessed only by karyotyping, and it's a very nice system or a model to explore the utility of optical mapping. And traditionally, another point I want to make is traditionally MDS is considered to have mostly copy number changes and less frequently translocations. But of course, there are some critical translocations that, if identified in MDS, implicate very poor prognosis. The problem is these translocations, they occur in a cryptic setting, and they're usually non-complex karyotypes. So oftentimes we miss that unless there is a really strong suspicion to do a specific targeted FISH on it. And these include the EVI1 or the MECOM rearrangement and MLL rearrangements. And MLL rearrangements are particularly common in therapy-related patients. By that I mean patients who have received prior chemotherapy for another cancer. And the second point I wanted to make is certain types of acute myeloid leukemia, which we call core binding factor leukemia. These can occasionally present with low blast count and along with dysplasia, leading to a misdiagnosis of MDS. The reason it's important to identify these core binder factor leukemias is that the treatment is completely different. So these patients respond very well to cytarabine and don't respond well to MDS related treatment. So it's very important. So if we have a system, a single platform assay that if it's able to identify both the copy number changes and the translocation at the same time, it would be very valuable in this setting. So in order to address that, we performed a pilot study, And we had two different questions. So the first question is optical mapping or whole genome imaging able to identify the structure variants that we are able to detect by the current standard of care assays, namely conventional cytogenetics, FISH, and microarray? And the second question is, since whole genome imaging is actually de novo assembly of the cancer genome, and it could potentially uncover cryptic translocations or cryptic abnormalities, is it able to provide new information beyond the standard of care assays that could potentially impact treatment? So I'm just going to go over a brief methodology that we used. So for the input, we used a high molecular weight DNA that was extracted by the protocol from Bionano. So we used two separate types of input material. One was frozen cell pellets, and the other was bone marrow mononuclear cells that were preserved in DMSO. And based on prior studies, Bionano had determined that the minimum cell number for successful mapping was about 1 million cells, and that yielded a DNA of at least 36 nanogram per deciliter. Now, at the time of analysis, the results were blinded, and for comparison, for actually calling out the somatic variants, we used 200 healthy controls that had been mapped before in the Bionano system in order to facilitate the detection of variants more than 500 base pairs. And based on, again, prior sensitivity studies, the LOD was determined as about a variant allele fraction of about 10%. So here's what we used. We used 18 bone marrow samples. 10 of these were live cells in DMSO and 8 frozen cell pellets. Now, all of the 10 cells, the 10 live cell samples gave us successful results. All had over 1 million cells and significant amount of DNA. On the other hand, for the frozen cell pellet, 2 samples did meet the cutoff of 1 million cells, and both of them gave us the successful results. In 6 samples, the number of cells were not sufficient, but we still tried to play with it, but those were not successful. So across the board, if we used the predetermined cutoff of 1 million cells and 36 nanogram per deciliter DNA, pretty much all the samples were successful. So to address the first question, first and the second question, I'm going to first illustrate a couple of cases to you, the potential of Bionano. So here is the first sample. This is a sample of MDS with 2% blasts, and by conventional karyotype, the patient had a del5q abnormality and AD12P13. Now, del5q abnormality is a pretty frequent abnormality, a recurrent abnormality in MDS. So here is the circus plot generated by optical mapping, and here is chromosome 5, and we see, if you look at the copy number track here, you see there is a clear-cut deletion in chromosome 5. In addition, there is a structure variant here that is showing up as a fusion between the remaining fragments of chromosome 5. So here we zoomed into the copy number track, and we see that there's a deletion from chromosome breakpoint 5q11.1 extending up to 5q35.2. And here is what we see. This is the SV track. So here what we see is actually the alignment of the genome fragments to the reference. So what we are focusing here is the fusion that we saw here. So here is, based on that, we can see that the fusion extends from this point in the reference until that point. So basically it is indicating that it's a second line of evidence that shows up, that's showing up that there is indeed a deletion of chromosome 5. So we can actually zoom in even more and understand this at the gene level. In addition to several genes that have been deleted, we also see that this particular gene, it has been disrupted based on this deletion. So in addition to deletion of chromosome 5, here is a second abnormality that we see. There seems to be a translocation from chromosome 1 to chromosome 12. In fact, there seems to be more than just a single translocation. And in addition, there's a duplication on chromosome 1 as well. So we can actually look at the plots between just the chromosome 1 and the chromosome 12 here. So we can, in the copy number track, we appreciate that there is indeed 2 translocations between 1 and 12. And there is a duplication, you can see here, of the chromosome 1 with an additional deletion. And there also seems to be a subtle deletion in chromosome 12. So here in the copy number track of chromosome 12, that confirms the deletion of chromosome 12. And if you look at the 2 fusions here on chromosome 12 again, zooming in into 1, we see that here is the transcript, the fusion transcript that's been generated. One fragment maps to chromosome 1, and here is the other fragment that actually maps to chromosome 12. So there is a fusion of 112, but in addition, there is this additional fragment that's mapping to neither of those. So upon further investigation, it was identified that this fragment actually mapped to chromosome 5. So it's indeed a derivative chromosome, a complex derivative chromosome. And this was not picked up by, I guess the resolution was beyond the limit of the karyotype and could be picked up by optical mapping. Of course, rearrangements such as these will be missed by microarray technology as well. So here is a second case. So this was a therapy-related myelodysplastic syndrome in a patient with a prior history of breast cancer, again 2% blasts. And conventional cytogenetics showed translocation 911, and there was another deletion 20q. So here is the circus plot again, and then the deletion 20q, we are able to appreciate it here. And there is the 911, and there seems to be another fusion. We can look into it now. So here is chromosome 20, the deletion 20q, the copy number track confirming this deletion here. And you can see that the deletion happens to be not uniform. There seems to be a subpopulation of cells showing a different set of deletions. So if you look at this fusion, we confirm that there are 2 separate fusions between within fragments of chromosome 20 that's consistent with deletions. And these deletions happen to be on different segments. Now this sort of heterogeneity in the population of neoplastic cells is pretty frequent in MDS and other hematologic malignancies. And it's actually implicated that these, the clonal heterogeneity is actually responsible for resistance to treatment and potential relapse at a subsequent time point. So we were very happy that optical mapping was able to indeed identify this heterogeneity in the neoplastic cell population. So, and then looking to the next translocation, so 911 translocation. Now the preliminary impression, if there's a 911, is that this patient is has a rearrangement of MLL gene, because especially in the therapy-related setting, that's the most frequent. But upon a closer view, we find that the translocation happens to be between 9p13.3 and 11q14.1, and here is the map. This maps to chromosome 11, and here is actually the chromosome 9 is flipped, and it's a translocation between 9 and 11. And in addition, there seems to be a deletion here. The deletion happens to be beyond the fragment, beyond the translocation breakpoint, as we can appreciate here, and the MLL happens to be within the deleted fragment and not related to the breakpoint. So after doing that, we confirmed it on FISH as well, that the MLL was indeed deleted and not rearranged, both by the FISH as well as karyotype mapback. So in addition to these two abnormalities that were also picked up by karyotype, there is one more fusion that we see between chromosome 4 and chromosome 7. And when we see, when we map it back here, here is what maps to chromosome 4, and this maps to chromosome 7. And there happens to be this fragment here that is mapping to neither, that in fact is suggesting a deletion, because it's not mapping to either. So if you zoom in further, so here is the reference chromosome 4. So this is the fragment of interest. A portion of it does map to chromosome 4, and the other point maps further down, confirming that there is indeed a deletion of this entire fragment. And when we looked into it, one of the most important genes implicated in myeloid malignancies, TET2, happens to be in that deleted fragment. Now, TET2 deletion is quite frequent in MDS. However, it is cytogenetically cryptic. So oftentimes, unless the lab is doing FISH routinely on all MDS cases for this particular deletion, we often miss that. And so here is, so the importance of TET2, either a TET2 mutation or a deletion, is that there are some papers that suggest that the presence of these actually suggests non-responsiveness to hypomethylating agent, and hypomethylating agent is now the standard of care for treatment of MDS. So this patient also had an 81 gene panel that we do routinely on all malignancies, and in addition to SF3B1, the patient has a frameshift mutation of TET2 as well. So to understand the whole pathogenesis, there's a simultaneous TET2 deletion and a frameshift mutation, so suggesting that TET2 is likely to function as a tumor suppressor here. So here is another case of MDS with a diploid karyotype. The circus plot is actually showing a subtle deletion on chromosome 11, which we indeed can confirm by zooming in here. So here is the map, and this maps to chromosome 11, suggesting that there is a deletion of this fragment. Now, what's the importance of this? Identifying this deletion. So as I mentioned before, the cytogenetic risk stratification and the revised IPSS score. So the deletion 11q happens to be in the very good risk category, as opposed to normal karyotype that is good. So as you can see, these have quite different survivals. So actually this was cryptic by cytogenetics, but optical mapping can identify this abnormality. So potentially the IPSS risk score of this patient is different. So overall, we as a pilot, we mapped 10 samples with a total of 38 aberrations that were known from prior conventional cytogenetics. And of these 10 samples, we used 7 with known cytogenetic aberrations. We also used 3 samples with normal karyotype. And out of these, 37 of 38 aberrations that were previously detected by conventional karyotype were also confirmed by optical mapping. And all of these cases also had microarray testing performed, and all aberrations that were detected by microarray could also be confirmed by optical mapping. The only aberration that was identified by karyotype but not by optical mapping or microarray was the deletion 7q. And it's because this happened to be, by FISH, we found that this happened to be in about 7% of the cells, and that's below the level of detection by both microarray and optical mapping. So based on our pilot study, we concluded that there is indeed a high concordance between optical mapping and the currently available standard of care diagnostic assays. And it's fairly, the limit of resolution of 10% is pretty, we think it's pretty good compared to the traditional conventional karyotyping. And at the same time, it's a high throughput technology that we could potentially use for clinical care. And as a result, based on the findings from this pilot study, we are expanding our study to perform validation to assess optical mapping as a single platform tool for routine clinical diagnostics. And we are going to expand this number of samples. We're going to expand the study to prospective samples that will be collected at MD Anderson. And we're going to include about 100 newly diagnosed myelodysplastic syndrome cases with a variety of cytogenetic abnormalities that include both copy number changes as well as translocations. And we will also include a few cases with normal karyotype to potentially uncover cryptic aberrations. And we will try to identify among the results, in addition to assessing concordance, we will also try to identify any genomic aberrations that could potentially alter the risk or change the patient management. And we also, hopefully, we plan to include profiling of these patients into the ongoing clinical trials. So overall, based on our experience, we do believe that the optical mapping can potentially be used for routine clinical diagnostics. And secondly, optical mapping has the potential to uncover underlying cryptic aberrations or even novel structure variants that may be potentially targetable and that could change therapy. Thank you.